Other thing which I want to add in terms of the analytical perspective, and most of the people in the field are aware of it, but each analytical technique brings some advantage, but also have limitations. So for example, when we are looking into the DLS, which is pretty much gold standard at the moment people use and FDA reporting, it gives you certain information. And then you go on a TM or a cryo-EM and you do the pretty much same kind of size analysis and you notice the difference.
Welcome back to part 2 with Jitendra Kumar of Entos Pharmaceuticals. We're picking up the conversation on proteolipid vehicles and the fusogenic platform, moving deeper into the practical and strategic questions behind delivering genetic medicines with a truly novel vehicle. Expect candid perspective on process, analytics, regulatory strategy, and what this technology could mean for patients where existing approaches fall short. Let's get back into it. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Another part that's absolutely vital is the regulatory part. Now, your platform is novel. It's a novel delivery mechanism, unlike lipid nanoparticles or, let's say, AAV. How do you approach the regulatory side and having these discussions with agencies? What is your strategy there?
As a company, our strategy was pretty much let's science talk. Went with all the data, all the informations, what we have. And obviously, you have to have your GLP tox study there. They are well aware where we stand for. In terms of the process also, you have to come up with say for our example, we had this membrane protein. So what are the limitations, what criteria we are taking, and how we are ensuring that the quality is maintained for a safe use. My take on the discussions with the regulatory, especially with the health Canada, is that they are open to have a new technology. They want to have a new technology in the fields. They promote those efforts. Their job is to make it safe. And if we ensure that we are matching all the criteria which are in the guidelines, and if not, we have a scientific explanation for that, how it is and why it is still valid. Because we are the one who are coming with the technology, we are having a scientific knowledge. So it is science-driven. We have to inform them in a proper manner. Your science has to talk bolder than what you want to say there when it comes to the regulatory.
And does this also include a solid analytical characterization package?
Of course. I mean, you have to have a good analytical package. That is where you are ensuring that you are meeting all the criteria.
And how different is that package for a fast protein than perhaps to an antibody or some more standard recombinant proteins?
The thing is that the fast protein is not our API. Our API is still a nucleic acid. It's one of the components on the surface of our particle. Having said that, we still have to meet all the criteria until it became a part of the PLVs, where we have to still show that is the presence, which is driving the delivery. So there are certain assays which we have to perform to ensure that there is a presence of protein on the surface and ensure that stability of the protein itself is a different thing, because that comes down to as a excipient, because it's a part of envelope, not the core of the DP or DS, where our API is a nucleic acid here. Having said that, we have done all the studies like making the viral clearance and everything to ensure that the protein is still safe and meeting all the criteria as we do for any other protein.
So that means that even though your PLV is not the API itself, you still have to have an extensive characterization for that because as you're delivering or inserting that into a patient.
Yes, we have to. And it's pretty much coming true for all the novel lipids as well, where I think FDA is asking to have a full characterization on the novel lipid which are not in their database, and which is fair ask. So anything which is even you consider as an excipient but is novel, they want to have more information there.
Now, I'm curious, Jitendra, you have made quite some progress. What do you still have to do in order to get a full approval, and how long will that take?
Obviously, the phase 1 and phase 2 are relatively shorter ones. At the moment, our product, which we think of getting into the market, which are most close at the moment, is a leptin therapy, which is for the lipodystrophy. We have to start phase 1, which we are hoping to start early next year. And then going for the BLA, probably it will take another 8 to 9 months at least. As you said, that's what are the different things which we want to address. And that's where we started to, the Cytiva bioreactor experiment was part of it. So that's how do we scale up? Is there a loss or can we enhance the products? And we all were addressing in terms of the scalability option there. In terms of the BLA options, obviously we have to go much more robust in terms of our analysis. And there are certain advantages, I will say, with the membrane protein. At least one of them is we have to maintain in a detergent, which is kind of a natural barrier for any viral to survive. So that gives us advantage. The disadvantage in that case, because for others, we have to do one of the physical treatment can be the detergent treatment. But in case of the membrane protein, they require a detergent to stay functional in the micellar space. I mean, most of the things we have for IND is applied to the BLA, but it's just a much more detailed studies which we have to complete and more stronger analytical. One more thing which I forgot to mention is that's also finding out the new technologies which can support you in that filing. And one of the work which we have done with the NIS in last year is that using the cryo-EM to characterize our particles. And it is a fantastic technology. You can visualize many things there. You can see cargo there. You can have your lipid layers. And one of the aim which we have there before BLA is to characterize the fast protein on the surface as well, which will give you proof that this is the one which is driving the whole technology visually. And coming back to my experience, so the time when I joined the company, one of the tasks which was assigned to me was also to the EM, which I got trained and did myself a lot of AM, TM and AFM just to get the particle size and architecture there. So other thing which I want to add in terms of the analytical perspective, and most of the people in the field are aware of it, but each analytical technique brings some advantage, but also have limitations. So for example, when we are looking into the DLS, which is pretty much gold standard at the moment people use and FDA reporting, it gives you certain information. And then you go on a TM or a cryo-EM, and you do this pretty much same kind of size analysis, and you notice the difference. You notice few things, and that is not because there is a size difference. The thing is, in one you are measuring the hydrodynamic radius. On the other system, for the EM, you are having a grid, and on grid, particles are getting dried, and because of that dry, they shrink in the size. So when you are comparing, you have to be aware what advantage or disadvantage any techniques is bringing to you as well.
Yes, this is absolutely key, and there is often not a one-size-fits-all strategy or... Quick pause before we get back to it. CMC is not something you deal with later. Treat it as an afterthought and the bill comes due. A stalled tech transfer, a $2 million rework, or a Series B stuck in due diligence. This one is for the founder heads down on the science and the CMC leader watching decisions get made without you in the room. I broke down the 5 recurring CMC mistakes that put promising programs at risk in a free 5-day email course called the CMC Failure Chain. The link is in the show notes. Now back to the episode. Method. I'd like to circle back to your career path. You have spent significant time on unmet medical need in oncology, neurodegeneration. So how does this now apply to what you're doing today, developing PLVs? What are these offering to perhaps new patient populations without any existing approaches or any treatment options?
I will start my answer from a different space. Long back, actually, first time when I joined as a summer student in India, I went to an institute called Indian Institute of Pulses Research. They work on lentils, and I was working in a lab which is doing a tissue culture. And it was great experience. I was there for 2 months. The head of the lab, Dr. N.P. Singh, I spoke to him, and I said that doing a lot of work here, and I don't see that much publications out of the lab. And one thing which he said was that we are working here for the drought-resistant varieties to develop for the areas, particularly in that state. And his response was that publications matters, but what matters is the real impact. And what we are doing is a real impact to those farmers who need crops to have a good yield, even in a drought area. And that is something which I feel I carry unconsciously everywhere. That's a lot of things matter, but ultimately it's how you contribute to the humankind, how you are impacting. Now, coming back to my career path and my journey, I have been lucky with, as I said, I work with the Herald. He's an amazing scientist and he's doing a lot of good work in the NMR spectroscopy. And that is where I started to work on an unfolded protein. One of my projects started as looking into the residual structure of the prion protein. And the concept behind that project was that since this protein mysteriously get unfolded and then misfolded within the brain, there will be some residual element within the protein chain which might be driving. And if we can identify those residual points, they can be defined as a target sites for the drug interaction. It turned out to be that even though hypothesis was good based on a few other protein which we have studied in the lab previously, even in a highly unfolded space, the prion protein aggregates. So my project turned into, rather than studying the residual structure, into identifying the unfolded state to the misfolded state. And I utilized NMR spectroscopy along with the EM, Max Planck Institute of Biophysics there. And I was lucky to interact with those people. Werner Kulbrand is one of the biggest name in the field of the cryo-EM and EM. So that's my journey there. I got interested in all the structural biology, protein-protein interaction, small molecule drug interaction. And a lot of people, they were working on a different project. And we had an industry partner there as well. So it's a very good experience I had in Frankfurt. However, that also got me curious about the biology of it, which led to my postdoc. And I came to Edmonton, where I am still staying and made it my home. And that was a different experience working with a neurologist, because then now you get a medical perspective of the disease. You are working on a disease where you are looking into the biology of it. And the project I was working on was basically isolating the infectious particle from the infectious brain. So that we got the brains from not from the human, but one is a prion disease, which is happening in a deer and elk population. It's called CWD. So we were getting those brains and we were isolating the particles from those and looking into the animal models for the infectious nature. And the whole development of field fluorescence technology, I was pretty much involved there. Other project which I got there was also designing a molecule for the sequestering of A-beta aggregates. So pretty much on a similar track. And that was the project where I got more interested or more motivated that can be something which can go to the good use for the humans directly. The peptides which we developed was good for the sequestering the aggregates. And the aim was to get into the thronostic with that one. And I got involved in a few discussions with groups here where we hoped we can take it further. Unfortunately, it did not happen. And then I made a transition to a different lab for the protein drug interaction. So I had quite a journey. But ultimately, what I learned overall is that whether it's the protein structure, RNA structure, or DNA structure, they are the key factor to drive many things. And one of the things which I try to address in my current role is also, I will say that one of the key challenges is that particle size. So when you are delivering any of the LNP particles or any nanoparticle, it has to travel within the body. And that travel depends on the diffusion rate. And the diffusion also depends on the size of the particles. So if you can make a smaller particle, it's going to be much more faster. And to make particle smaller, you have to know the packaging of your mRNA, for example, how it's folding and how can you make it more compact so that your envelope is also tight on it. And then also to add on that one, the new layer of the research and everybody pretty much jumping on that one is having a corona layer on the particles, which allow you to have a targeted delivery. But that is where my previous knowledge comes in a picture where I can utilize that to design these particles or help in designing these particles.
This has been great, Jitendra. What additional questions should I have asked?
I mean, I haven't listened to your podcast. It's mostly on the CMC, and I have done the CMC part, but mostly like my major role is regarding this membrane protein. So I can talk about it, but it's pretty much similar there. Having said that, the biggest thing is that it allows easy fusion or stable direct fusion to the cell membrane, which people do not realize that there can be a method where your particle can fuse with the cells and deliver directly to the cytosol. Regarding questions, I don't know, to be honest. It's up to you. If you have anything in your mind, anything, I mean, in terms of the work-wise, as I said or mentioned, that peptide which I designed for the sequestration, that is close to my heart. If I get a start capital, I will work on that one because having exposure to the industry, I know the drawback now. Because the problem is with any neurodegenerative disease is diagnostic. The time which we find out is disease is already progressing into the state where it's hard to reverse. But having said that, that peptide, which I worked in 2013-14, was pretty good in sequestering the A-beta aggregates. So the way I see is that if we have a, say, for example, Foley labeled, which we can do with the MRI, we can do the early diagnostic. And the advantage it is going to be that because it sequesters and actually clears the particles. So not only you are finding out the rich aggregation, but if anything happening, you can clear it. But in terms of the industry, what I found out is it was not that exciting for the people unless I get into the animal model and give a proof of concept, which involves a lot of money. So there are limitations everywhere.
You're right. And early detection is key. And hopefully we'll find ways to make that happen.
And the other thing, I mean, whether as a question it can be or not, but the difference between academic and industry setup is that to identify where to stop. A lot of times exciting things will come, but you have to make a hard stop just because what is feasible for your company or what is feasible for your time and efforts. There are a lot of scientific curiosities. There can be many paths which can lead to the maybe same results, but you still have to define what is your path there, especially when we are working, say, for even with the technology where we change few compositions here and there and we get a mixed results. We still have to decide on one.
Yes, focus is key.
Yeah.
What is the most important takeaway from our conversation, Jitendra?
In my opinion, finding the right partner, having good network, and be aware of strength and the weakness of your technology. As much as importance is to know the strength, you have to find out your weakness as well, because that is where you have to address them as soon as possible and make it stronger.
Excellent. Where can people get a hold of you and learn more about your technology?
I mean, people can reach out to me on my LinkedIn and people can reach out to entosPharma.com. That is where we can respond to the technology much easier way.
There you got it, smart biotech scientists. I will leave the links in the show notes. Please reach out to Jitendra and his team. Thank you so much, Jitendra, for being on the show today and sharing your passion and making a difference in drug deliveries. Thank you so much.
Thank you, David. Thank you for the invitation. It was a pleasure to talk to you and share my journey.
Novel delivery platforms demand new playbooks. Jitendra Kumar has shown how process decisions, analytical strategy, and regulatory thinking come together when the precedent simply isn't there yet. From phase 1 trade-offs to the question every CMC leader evaluating non-viral delivery should be asking. A masterclass for anyone building in this space. Thank you for tuning in today. If this episode was useful, please leave a review on Apple Podcasts or your preferred platform. See you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.